Right, so like I was alluding to earlier, what we can do is we can take a cohort of, let's say, 50 triple-negative breast cancer PDXs that we did in this study, and we basically treated the PDXs with single-agent carboplatin or single-agent docetaxel or their combination. And in that regard, we can get chemotherapy responses to the single agents and their combinations. And from that, we can also profile the tumors. Basically, what that means is we can look at all the genes, measure them by whole exome sequencing, RNA sequencing, and at the protein level with proteomics. We can basically relate the molecular profiles now to the treatment responses that we saw to the single agents and their combination. And through all of these association analysis with our diverse molecular profiles together with chemotherapy responses, we're able to find diverse sets of genes and distinct sets of genes that were associated with single agents to carboplatin and docetaxel versus their combination. And these were largely non-overlapping, so the genes that are associated with single agent responses are not the ones that are associated with combinations, and the ones associated with carboplatin response are not the one associated with single agent taxane responses. And the other thing that we did very importantly was we generated chemotherapy predictors to predict chemotherapy response using our PDX system, together with publicly available databases with patient data that had been similarly treated with similar chemo regimens. And from there, we were able to predict in a very accurate and specific manner responses, or we were able to identify biomarker combinations at the mRNA level that were predictive of single agent responses and their combination.